The American diet is incredibly high in copper. Nuts, peanut butter, avocados, arugula, on and on we go. Everything has high copper in it. We're just not, most of us aren't genetically capable of handling that much. If you tell a fibromyalgia patient or your mom or your friend or cousin, get on a low copper diet, they'll improve 80%. So American diet plus NAT2 SNPs plus glutathione SNPs plus MTHFR SNPs equals fibromyalgia. I have a cohort of 7,000 patients in fibromyalgia with natural options, my Facebook page on this. Go look at it. And they get better. They go from bedridden in agony and pain. They get on a low copper diet. It's a copper toxicity issue. It's what it is because of those SNPs. NAT2 causes ceruloplasmin dysfunction. Well, ceruloplasmin is how you move copper around and zinc around in your body. NAT2 SNPs, because NAT2 you need boron, molybdenum, stuff like that, B6, all that for it. We figured that out after we'd gotten a patent on our copper balance, which has all that in there. This is just really a ceruloplasmin support product. For the people that have NAT2, they suddenly get better with their fibro, and we get people who've gone from a level 10, or like my wife said she had a 14 for her pain because we'd been married for a while, and she told me she had fibro. I was shocked. I'm an MD, and most MDs, please excuse my French, we're dicks. I mean, we're kind of snippy and mean, and we think everyone's got a drug addiction problem, stuff like that. So that was my thought on fibromyalgia. She took ibuprofen occasionally, but apparently she took a lot of it. And I swore I'd try to figure out what caused it. Naturopaths had figured it out. They figured out they give people molybdenum or boron, they got better. They couldn't figure out why they got better, but they knew they did get better. And I'm the one who went, why does that make them better? And then I realized when I kept seeing NAT2 problems come upon all these fibromyalgia patients, it's the boron and the molybdenum and the B6 and a bunch of other different things that are in our copper balance that cause your ceruloplasmin to be dysfunctional, so you can't move copper into and out of your cells. Also, the CMA or the SpectraCell kept coming up with copper deficiencies on an intracellular basis. Why was that? When they'd have super high serum levels of copper, why would they be low inside their cell yet super high outside their cell? Finally, I figured out it's the ceruloplasmin. Ceruloplasmin was not transporting the copper into and out of the cells like it should.